Dear students, today we learn a technique by which we can determine the concentration or number of bacteriophages present in a given sample by plating method. This method is also called Plaque assay. So we learn the principle and procedure of Plaque assay, and at the same time the lytic life cycle of a bacteriophage inside its host. Viruses are obligate parasites. That means they are living inside the host cell but non-living when they are outside of the host cell. And viruses are very specific about their hosts. Like some viruses attack animal cells and are called animal viruses. Some viruses attack the plant cells and are called plant viruses. And some viruses attack bacterial cells and are called bacterial viruses. They are also called bacteriophages because phage means to eat, and since these bacterial viruses infect and kill and lyse completely the bacterial hosts, they are called bacterial phages. Bacteriophages were first discovered by Felix Stuart in 1915. Though the first evidence on bacteriophages came in the year 1896 by the scientist Ernest Hanbury Hankin. He noticed some factors are present in the rivers of Jamuna and Ganga in India which had antibacterial action against cholera. And in 1917, Felix de Herel independently discovered bacteriophages. And this time he discovered the bacteriophages against Bacillus dysentery. And he treated a person with these bacteriophages and cured that person. Thus, he introduced the phage therapy technique. Now, we know that T phages of E. coli are lytic phages. Do you know how many T phages have been discovered so far?
7.
7. Good. 1 to 7. 1, 3, 5, 7. These 4 viruses are called T odd phages, and 2, 4, 6 are called T even phages. All of them infect different strains of E. coli and follow lytic cycle. Now, this is the picture of common T even bacteriophages. T2, 4, 6 belong to the family Myoviridae. The Greek word myos means muscle, and since the tail is contractile, it resembles muscle, and thus they belong to this group Myoviridae. Bacteriophages are very specific about the hosts. All T phages can infect different strains of E. coli, but T4 infects very specific hosts like E. coli K12 and strain B. So, if we are performing the Plaque assay with T4 as the bacteriophage, then we have to take its specific host, either K12 or strain B. Now, T4 bacteriophage belongs to the Baltimore group 1 since it has double-stranded DNA as its genome, and it belongs to the order Cordovirales and family Myoviridae. T4 has been used as a model system by many renowned scientists, and it has served as a very good model for the study of genetics and molecular biology since 1940. This is the structure of T4. This complex capsid is composed of a big icosahedral head which is multiprotein, a neck, a contractile tail, tail fiber and base plate, and this is a very large lytic phage. And if we look into the genome of T4, it is terminally redundant and circularly permuted. The genome is linear double-stranded DNA of about 169 kilobase pairs, and it has terminal redundancy. That means a sequence of 1.6 kilobase pair is repeated at both the ends. Now, if we look into the progeny viruses coming out of an infected host cell, then we will see all the genomes are terminally redundant. That means in all the genome, 1.6 kilobase pair is repeated at both the ends, but the sequences vary among the progeny viruses. That means the population is circularly permuted. Now, since it is a lytic phage, it always follows the lytic cycle, and if we look into the timing of the life cycle of T4, then just after adsorption to the phage, it injects its genome into the host cell and turns off the synthesis of RNA, DNA and protein of the host origin. The first mRNA of viral origin synthesis starts around 2 minutes after infection, and late mRNA synthesis begins at around 9 minutes after infection. And at around 12 minutes, we can see complete head and tails inside the infected host. and at around 15 minutes complete phage appears inside the host. And when many phages accumulate inside the host, they will lyse the host cell and come out at around 22 minutes. Now, if we look into the details of the infection, then first step of infection is adsorption of the T4 to the bacterial surface and injection of its genome. Now, T4 uses bacterial cell surface proteins and polysaccharides as the receptors. The tail fibers contact the receptors, the base plate settles down on the bacterial cell surface, the sheath becomes wider and thicker, and the protein attached to the base plate has lysozyme-like activity. It lyses the bacterial surface and the tail is injected into the membrane, and the DNA which is present inside the head is injected inside the cell. The T4 genome carries almost 300 genes and they are under 3 different promoters: early promoter, middle promoter and late promoter. Now, a protein comes with the genome from the T4 head inside the E. coli cell. That protein modifies the host RNA polymerase in such a way that the host RNA polymerase no longer recognizes its own promoters. Rather, it starts transcription of the viral genome. Okay. Middle promoter controls the expression of the genes which are necessary for the replication of the T4 genome, and the late promoter controls the gene necessary for synthesis of the structural proteins. Once the T4 genome has been replicated and structural proteins has been synthesized, assembly process will start. The structural proteins will form the capsid, the replicated genome will be inserted inside the icosahedral head and the complete phage will be produced. And when about 300 new phages are produced inside an infected E. coli cell, they will lyse the host cell. After lysis, if the newly released host cells get new E. coli cells in the neighborhood, then they will attack the new E. coli cells, okay, and they will grow inside the new E. coli cell. Again, they will lyse the E. coli cells, okay. And in this region, no host cell will be present. All the cells will lyse due to the infection by T4. So, a clear region will be produced. These cleared areas are called plaques. So, in a bacterial lawn, we will see cleared areas called plaques. And the characteristics of plaques, that is size and shape of the plaques, depend on the particular bacteriophages and also on the physical and chemical characteristics of the medium in which the bacteria are grown. And also it depends on the incubation time. Theoretically, if one bacteriophage infects one host cell and grows inside it and then lyse that host cell and infect the neighboring cells, then one plaque will be formed. So, counting the plaques, we can calculate the number of phages present in a given sample. Though in practice, we represent the result as plaque forming units. That means number of virus particles capable of forming infectious plaques. Plaque assay was first discovered by Felix D. Herrell, and many scientists then worked on this technique and developed the technique. And plaque assay is mainly used to determine the concentration of bacteriophages present in a given sample, also for isolation and purification of the bacteriophages. It will take almost 3 days. On day 1, we have to inoculate 5 ml of nutrient broth or peptone broth with a single colony of E. coli K12 strain or E. coli B strain and incubate it overnight at 37 degrees centigrade. On second day, you take 1% of the overnight grown culture and inoculate 25 ml of tryptone broth containing tryptone and calcium chloride in it, and incubate for about 3 hours at 37 degrees centigrade till the OD reaches at 0.6 at 600 nanometer.
Ma'am, why do we add CaCl2 in the media?
The presence of divalent cations help in the adsorption of the T4 to the bacterial host. So, it facilitates the adsorption of the bacteriophage to the host and penetration of its genome.
Ma'am, why do we incubate it till the OD reaches 0.6?
OD of 0.6 at 600 nanometer is an indication of the log phase culture. That means all the E. coli cells are growing vigorously and multiplying. So, they are in the log phase. So, very quickly they will be infected by the bacteriophage. The cells are centrifuged at 500 rpm, that is rotations per minute, for 10 minutes in sterile centrifuge tubes, and cells, since they are heavy, they will come at the bottom of the tube. So, we will discard the supernatant, we will collect the pellet. Now, we will add few ml of broth to the pellet and resuspend the pellet. Now, we will dilute the phage sample. Since we do not know the concentration of the phages present in the sample, we need to dilute it manifold. Okay. So, to dilute it, we have to first take 10 test tubes or 10 tubes, and we will take 900 microliter of tryptone broth in each of them. Now, from the phage sample, we will pipette out 100 microliter and we will add to the first tube. Okay, and we will mix it properly, and the dilution becomes 10 to the minus 1. Then from this 10 to the minus 1 dilution, we will again take out 100 microliter and we will add to the second tube. Again we will mix properly, the dilution will become 10 to the minus 2. Again from the 10 to the minus 2 dilution, we will add 100 microliter to the third tube and the dilution will become 10 to the minus 3. This way we will continue diluting the phage till the dilution becomes 10 to the power minus 10. In the meantime, we will prepare 10 tryptone hard agar plates and 10 sterile tubes labeled from 10 to the minus 1 to minus 10. Now, we will allow the E. coli cells to be infected by the T4 phage. For that, we will take 100 microliter of plating cells and 10 microliter of different phage dilutions and we will mix them separately. Now, after mixing, we will keep them at 37 degree centigrade for 15 minutes. This time is given for the adsorption of the phage to the host and injection of its genome inside the host. Okay. And during mixing, you have to mix it very gently. Okay. Don't do vortex, because if we do vortex, then the phages will be separated from the host. Now, 100 microliter of phage host mixture will be taken from each tube after 15 minutes and will be added to 5 ml of tryptone soft agar. You know what is soft agar?
In soft agar, the percentage of soft agar is less compared to in normal media.
In soft agar, the soft agar concentration is normally 0.8%, and we will mix it between palms for 2 to 3 seconds. But remember one thing, you have to maintain the soft agar temperature around 42 degrees centigrade. It should not exceed 45 degrees centigrade or should not go below 40 degrees centigrade, because if the temperature exceeds 45 degrees centigrade, then the host cells may die, and if it is below 40 degrees centigrade, then the soft agar will be solidified. So, maintain the soft agar temperature at 42 degrees centigrade by keeping it in a water bath. The mixture of host phage with soft agar is poured immediately onto the respective hard agar plates. Hard agar means the agar containing normal agar, that is 1.5 to 1.8%. And as soon as the soft agar is solidified, the plates are incubated at 37 degrees centigrade in an inverted fashion. The E. coli cells has got the genome of T4. The T4 genome will be expressed inside the E. coli cell. New T4 viruses will come out of an E. coli cell. They will infect the neighborhood cells. That means the newly released phages should diffuse through the medium, and this is the purpose of soft agar. It allows the smooth diffusion of T4 phages through the medium. That is why we used soft agar. And also it eliminates the uneven absorption of host phage on the hard agar plate. Now, on day 3, we will observe and count the number of plaques, and the appearance of plaques depend on the phage concentration and also on the multiplicity of infection. It is a ratio of number of infectious virus particles to the number of target cells present in a defined space. Suppose in this example, the multiplicity of infection has been shown as 0.1. That means only one phage is present among 10 host cells. So this phage will infect this particular E. coli cell, it will grow inside the E. coli cell, it will come out and it will get new host cells to infect. So a clear zone will be observed, we will get a clear plaque. But if the multiplicity of infection is 1, that means number of phages and number of host cells are equal, then what will happen? All the bacterial cells will be infected by phages and they will be lysed. So complete clear area will be observed, not a particular clear area in a bacterial lawn. So we can't observe any plaque. So multiplicity of infection or MOI should be kept low. We have to count the average number of plaques present in a countable plate divided by the dilution factor and the volume of phage sample used in the experiment. Suppose we have got 13 plaques at a dilution of 10 to the minus 7 and we used only 10 microliter of phage sample. So the number of plaque forming units per ml is 13 divided by 10 to the minus 7 and divided by 0.01 ml. That means 1.3 into 10 to the 11 plaque forming units per ml. This is the result for a particular experiment. Here we can see cleared zone, okay, like an empty plate in 10 to the minus 1 and minus 2 dilution, because too many phages were present in 10 to the minus 1 and minus 2 dilution. So they lysed all the bacterial cells. Okay, no countable plaques are present in the plate. That is why complete lysis is observed in these 2 dilutions. In 10 to the minus 3, minus 4, again too many phages are present, but though the number is less than 10 to the minus 1 and minus 2, the plaques merge together and has formed confluent lysis. Okay, and we can't calculate the number of plaques from this plate also. In 10 to the minus 5, 1400 plaques were observed. Okay, so we can easily count the number of plaque forming units present per ml. In 10 to the minus 6, 800 plaques observed. So this is the plaque forming units per ml. In 10 to the minus 7, very few, only 13 plaques are observed in the plate. So this is the plaque forming unit. And in 10 to the minus 8 dilution, only 1 plaque appeared on the plate. So this is the plaque forming unit. So from all the observable or countable plates, we can see the plaque forming units are in the range of 10 to the 10 per ml. And in 10 to the minus 9 and 10 to the minus 10, complete bacterial lawn is observed, because very few phages are present to form any observable plaques. Maybe no phages are present in these 2 dilutions. Now, if we want to elute the phage from the plaque assay plate, what we have to do? We have to remove a plug of soft agar containing the plaques. Okay, then we have to place it in sterile saline water, and within 15 to 30 minutes, the phages will diffuse out of the agar, they will come to the saline. Okay, then a rapid spin, the agar will be precipitated out and the phages will present in the supernatant. So you collect the supernatant, it is full of T4 phages. Okay, you can keep it at refrigerator indefinitely. While performing the experiment, we need to remember these few points, that you remove the soft agar one by one from the water bath, otherwise it will be solidified. Okay, and don't do vortexing when you are mixing host and phage, and when we are pouring the soft agar plus host and phage onto the hard agar plate, the hard agar should be completely dry and no water should be present condensed on the hard agar plate. Okay, we can eliminate the condensation by pre-warming the hard agar plates in an incubator at 37 degrees centigrade. So just remember these things while you are performing the assay. We have learned how T4 propagates inside its host, the principle and procedure of plaque assay, and how we can interpret the results of a plaque assay.